Guys, welcome back to my lectures. In today's lecture, we are going to see Hershey and Chase experiment of transfection. We have seen Griffith's experiment showing transforming principle to show genetic material, and the second one was we have seen Avery MacLeod and McCarthy experiment, which also shows that DNA act as a genetic material, which was the first experiment carried out, and the links for these both two videos will be uploaded in my description box. Please do check out. Now let's see the transfection. Hershey and Chase works on the bacterial infection which was occurred by bacteriophages. That means bacteria were infecting bacterial cells. So the transfection was termed came into existence. That means it is the gene transfer method in which naked DNA or RNA introduced inside the bacterial cell or eukaryotic cells by chemically. That means it is the introduction of bacteriophages into the bacterial cells. So this is nothing but the transfection method. These experiments or Hershey and Chase used experimental tool, that is T2 bacteriophage. Why they use T2 bacteriophage? Because these bacteriophage contain protein coat and nucleic acids. These two parts are the major components of these bacteriophages. Thus these T2 bacteriophages were used for their experimental work. So this is the these are the body parts of the bacteriophages. That means they have collar, they have whiskers, they have sheet, they have basal plates for the attachment, they have tail fibers, and the spikes. Spikes will introduce the bacterial DNA inside the bacterial cells. Okay, and this is the outer protein coat. So this is the structure of T2 bacteriophage. Now these experiments use the technique, that means radio labeling techniques was used. Now what is mean by radio labeling? Radio labeling is nothing but the procedure where some known atoms are replaced by their radioactive isotopes. That means we know that proteins or any biomolecules are made up of atoms. So during the radioactive labeling, these atoms are replaced by their radioactive isotopes, the specific radioactive isotopes. Okay, then these replaced atoms or these replaced radioactive isotopes will emit radioactivity and hence work as labels to mark molecules of the interest. So this experiment used radio labeling of proteins and DNA because they wanted to check either proteins or either DNA acts as a genetic material. Thus proteins were labeled with radioactive sulfur 35S in place of regular sulfur 32S, because protein molecules are made up of building blocks of amino acids and they are made up of sulfur molecules. Thus proteins were labeled with 35S radioactive sulfur and DNA was radioactively labeled with 32P phosphorus, because DNAs are made up of phosphorus molecule. Thus proteins were radioactively labeled with 35S and DNA was labeled with 32P, that means phosphorus. Clear? Now coming to the experimental part, two experiments were performed. In first experiment, testing of proteins were done, and in second, testing of DNA was done. So in testing of protein, the protein coat which is present outside the bacteriophage, was labeled with 35S phosphorus, right? Then the infection was done on bacteria, but no radioactivity observed because no radioactivity enters inside the cell, because these are the protein coats. Okay, bacteriophage will emit their progeny and they were carried outside the bacterial cell, but no radioactivity enter the cell. And when phages were grown with radioactive sulfur 35S, after centrifugation, radioactivity was observed in supernatant, not in the palate, because after centrifugation we get two parts, that means supernatant and palate. But radioactivity was observed in supernatant because proteins are the laser dense molecules, thus will reach in the supernatant part, but not in palate. Thus radioactivity observed in supernatant. So in second experiment, testing on DNA was carried out. Here DNA was radioactively labeled with 32 phosphorus. Then infection was done on bacteria. Radioactivity enters inside the cell because DNA will be carried inside the cell, thus radioactivity enters the cell. When phages were grown with radioactive 32P phosphorus, after centrifugation, what we get? We get radioactivity was observed in pellet, not in the supernatant form. Thus it was concluded that proteins are not genetic material, but DNA is the genetic material, because DNA is DNA is passed into the bacterial cell, but proteins are not passed into the bacterial cells, that no radioactivity was observed inside the cells. Understood? This is the easiest flow diagram of Hershey and Chase experiment, where you can see the phage particle where protein codes were labeled with proteins. Then infection was carried out, and after the blending, no sulfur detected inside the cell, but sulfur detected in supernatant. Okay, so the cells means pellet considered, and supernatant were not showing the presence of sulfur. But second, you can see the phosphorus containing DNA was labeled with 32P phosphorus. Then infection was carried out. After blending, phosphorus were detected inside the cell, because DNA is carried out inside. DNA will carry inside the cell. Thus, no phosphorus detected in the supernatant, and hence it was observed that when radioactivity was measured in pellet and supernatant of both media, 32P phosphorus was found in large amount in pellet, not in the supernatant part. Thus concluded that DNA is carried or transferred from one viral cell to host cell, not proteins. Thanks for watching.